Good afternoon, dear students. Today we are going to discuss here our next experiment, that is experiment number 3, single radial immunodiffusion. Rather we can say radial immunodiffusion. What is it? Now see, immunodiffusion is a technique which is used for detection or measurement of antigens and antibodies by their precipitation, where the technique involves diffusion of antigens through a substance like through agar, okay, or through agarose gel. Or rather you can say that immunodiffusion is precipitation reaction in gel. What is immunodiffusion? Precipitation reaction in gel, where antigen and antibody react with each other and form a precipitation reaction, which can be seen in the form of a precipitin ring. Okay. So see, antigens and antibodies react with each other to form flocculation or agglutination. Here they react because the antigens are soluble, the antibodies are also soluble, they form precipitate. Okay, so here we have to see precipitation reaction. Okay, there are several methods of obtaining a precipitate between antigen and antibody. One of the method to obtain precipitation reaction between antigen and antibody is immunodiffusion. How can we do this? Okay, suspending the antigen or antibody in the gel and letting the other one to migrate through the gel from the well where it is placed. That means either antigen or antibody may be present in the gel itself. Like here in single radial immunodiffusion, the antibody, the antiserum is present in the gel. Now, antigens are placed in the well, and that means the patient sample containing the antigen is placed in the well, and only one thing diffuses out. What diffuses out is the antigen. So the name single. And because we are placing the sample in the well and the sample diffuses in all the directions, that is why the name single radial immunodiffusion. Okay. Now letting the antibody and antigen migrate through the gel from separate wells such that they form area of precipitation. Now based on what method you are applying, immunodiffusion may be radial immunodiffusion, and number 2, outer Loney double diffusion. See, in radial, suppose this is your plate, and on the plate we have placed agar gel and the agarose gel. Agarose gel contains antibody. That means antibodies are present in the gel itself. Now we have to dug out a well in the gel. We dug out a well, and in the well we place the antigen. Okay, so the antigen is placed in the well. So this is the well where we place the antigen, and the antigen diffuses out of the well in all the directions. That is why the name radial immunodiffusion. Now out of the two, the antigen and antibody, antibodies are already in the gel. Only one of the two diffuses, that is the antigen here. So the name single radial immunodiffusion. The other is outer Loney double diffusion. In case of double diffusion, two things move. Okay, both antigen and antibody can move. So immunodiffusion techniques can be radial immunodiffusion, a single radial immunodiffusion technique where antibody is put into the gel, antigen is measured by the size, and antigen present in the sample is measured by the size of the precipitin ring formed when antigen diffuses out in all directions from a well which is cut into the gel. In outer Loney double diffusion, both antigen and antibody diffuse from the well into the gel towards each other. Right. So here we are studying radial immunodiffusion. Radial immunodiffusion, RID, where we follow Mancini procedure, is also known as Mancini immunodiffusion or single radial immunodiffusion assay. It's a single diffusion technique whereby a solution containing antigen placed into the well in a gel or agar surface evenly impregnated with antibody. That means the gel is impregnated evenly with antibody. Antigen is in the well which is cut in the gel. The diameter of the ring that precipitates around the well as a result of antigen-antibody reaction corresponds to the amount of antigen present in the sample. Okay, now suppose this is a slide. We have cut a well and we have placed the antigen. The antigen diffuses out into the well. Okay, so the antigen diffuses out into the gel, and you can get a precipitin ring. Why do you get a precipitin ring? Because antibody present in the gel and antigen present in the well react with each other, form precipitation reaction, which can be seen in the form of a precipitin ring. Now, the diameter of the precipitin ring determines or depends upon the quantity or amount of antigen present in the sample. So this is a quantitative method whereby we can measure the quantity or amount of antigen present in the sample. The Mancini immunodiffusion test is carried out in one or more of the following objectives. What is our objective? To detect antigen antibody complexes, to detect or to find out the concentration of antigen present in the gel, to determine the concentration of antigen present in the sample, then describe the circumstances under which antigen complexes precipitate out. Rather, most of the time we study radial immunodiffusion to find out the concentration of antigen present in the patient's sample. What is the principle of radial immunodiffusion? It is a type of precipitation reaction. It is based on the principle of precipitin curve, which states that antigen antibody interact, forming visible cross-linked precipitate when the ratio of antigen to antibody, when the optimum ratio of antigen to antibody is present. Okay, so when an optimum ratio of antigen antibody is present, they react with each other, they cross-link and form a precipitate. In the test, antibody is incorporated into agar and poured into glass plate to form a uniform layer. So we take agarose gel, and in the agarose gel, we prepare agarose gel in a beaker, boil it, and into the gel itself we add the antisera or antibodies, and then the gel containing the antibody is poured on a slide, so that the whole antibody is present in the complete gel uniform concentration, and then we dug out circular wells into the agar. Okay, and antigen is poured into each of the well. Okay, now so we dug out wells. Specific antigen to the impregnated antibodies diffuse through the agar well in all direction and react with the antibody present in the gel, forming visible precipitate or precipitin ring. Ring-shaped bands of precipitates form concentrically around the well, indicating the reaction. The diameter of the precipitin ring form corresponds to the amount of antigen present. What is the procedure? Agar containing appropriate antiserum, that is antibody, is poured in glass plates. Now what you do, take a beaker, okay, prepare agarose gel, boil it to dissolve, okay, and in this agarose gel you are going to add antiserum containing the antibodies, right? And then this agarose gel containing the antibodies is poured on a flat surface of a slide. So what you have is a slide. Containing antibodies mixed with agarose gel. So gel containing antibodies. And then you dug out wells. Okay. And in the wells, what do we place? In the wells, we place antigen. In the well. And incubate, you prepare a moist chamber. That means you take a petri dish, place a wet cotton in the petri dish and place your slide. Otherwise the gel will dry. And then close the lid, incubate. Okay, during incubation what happens? The antigen present in the well will diffuse out in all direction. Okay, and it will react with the antibody that is present in the gel. And a visible reaction will occur between antigen and antibody, and the reaction can be seen in the form of a concentric ring called as precipitin ring surrounding the well. You have to measure the diameter of the precipitin ring, and by plotting a graph of diameter of precipitin ring versus concentration of antigen, you can find out the concentration of antigen present in the unknown sample. Okay, this is how we carry out the procedure. As the antigen diffuses radially, ring of precipitin will appear in the form of in the area where there is optimum antigen antibody concentration. The diameter of the ring is noted down. A standard curve is prepared. Okay, standard curve of diameter of precipitin ring versus the concentration of standard antigens. This curve is then used to determine the concentration of antigen present in the unknown sample, right? So we plot a graph on this. We plot on the y-axis, there is diameter of precipitin ring. Here on the x-axis, there is concentration of unknown antigen, right? Now, concentration of standard antigen. Concentration of standard antigen. You know the concentration of standard antigen. You know the diameter of the precipitin ring. You get a graph. Okay. Then, you know the diameter of the precipitin ring of unknown sample. Okay. Extrapolate that on x-axis and you will come to know what is the concentration of antigen present in the patient's sample. Right. So, this is how we place the gel. Wells are dug. Glass slide is coated with a gel containing antibodies. Then you dug out, say here we have dug out 4 wells. In the first, second and third well, we place the standard antigen whose concentration is known, and in the fourth well, we place the antigen from our patient's sample, right, where the concentration of antigen is not known. Antigen is added to the antibody rich media, okay, and then the antigen diffuses out, okay, antigen will diffuse out. And when it diffuses out, you can get a precipitin ring. You can see here precipitin rings. Now, what you have to do is measure the diameter of the precipitin ring, right? And then plot a standard graph like this. So, on y-axis, we have diameter of the precipitin ring. On the x-axis, concentration of the standard antigens. You know here. So, standard 1, 2, 3, you can see, right? And then, by plotting you can see, just suppose the concentration or the diameter of precipitin ring of the unknown is here. Diameter, mark the diameter and extrapolate it on x-axis and you will come to know what is the concentration of unknown antigen. Is that clear? So this is the diameter, say suppose, you extrapolate it and this will be the concentration of antigen present in the test sample or unknown sample. This is how you get antibodies in the gel and antigens in the well. Antigen diffuses out and precipitin rings are formed. You can see precipitin rings, right? You can measure the diameter and plot a graph like this and then find out the concentration of antigen. So what is the application? Immunodiffusion techniques are mostly used in immunology to determine the quantity or concentration of antigen that is present in the sample. And estimation of immunoglobulins in the serum can also be done, like estimation of IgG or IgM antibodies for various flu viruses can be detected by single radial immunodiffusion. Not just antigens, we can also detect the concentration of antibodies, whether they are present or absent, right? So the relative concentration of antibodies in serum, then various proteins, disease diagnosis, serological surveys can be done by radial immunodiffusion technique. So that is all about SRID. That is experiment number 3.